Thank you so much, Brian, and thank you for the invitation. This has been a really interesting meeting so far, fun to hear the different talks, and happy to contribute with perhaps a slightly different direction. So I'm going to be talking about a project, in fact, that Brian and I are working very closely on, and it's about trying to decode chemical effects at a single cell level. Can everyone see the slides and hear me?
Yes. Looks
Perfect.
good.
And is the background noise gone down a little bit?
Oh, it's fine now. It was just when you and Brian were both speaking, but all good.
Okay. So let me give you a little bit of context about what we're doing and why we're doing it. So as you all know, new approach methods are needed for evaluating chemical safety. High throughput NAMs, like a lot of folks have been talking about today, including transcriptomics, cell painting, high content imaging, they're really good at identifying pathway perturbations and estimating molecular PODs. But the question always remains, are we talking about adaptive effects, or are we talking about adverse effects? This is a problem that I've been working on with several colleagues for a few years. And the idea is that if we increase the level of a chemical beyond a certain critical point, can you cross some adaptive threshold that overwhelms the homeostatic capacity of the system, and does that lead to adverse outcomes? And I got to thank Katie Sadie for making a very wonderful graphic depicting this in a conceptual way. And the notion is that when you expose cells to various chemicals, if you don't exceed this tipping point, if the perturbations don't exceed this tipping point, you can have recovery. Whereas if you exceed this tipping point, you would have potentially irreversible damage. All right, so the questions that we're interested in is trying to figure out, are there in vitro surrogates of tipping points? Can we estimate the critical concentrations for chemicals at these tipping points? And then what are the molecular mechanisms and signatures of tipping points so that we can build better tools, in fact, to identify tipping points from NAM-based data. So these are some of the issues that we're interested in working on in the EPA Virtual Tissues and Tipping Points Research Project, which has two main components. There's an in vitro component that involves organotypic cell culture models, where we are looking at either physiologically relevant systems or organotypic models for capturing the different heterogeneous cell types that exist in tissues and make them resilient to environmental stresses, and also looking at in silico approaches such as computational systems models that can simulate the mechanistic relationships using NAM-based data to predict traditional outcomes or tipping points. And we're in this project also looking at cell-level data streams to enable systems modeling that could distinguish between adaptation and adversity. Going to slide number 3. So previously, we've actually been working on this project for quite a while. The first project that we worked on looking at analyzing tipping points was based on the notion of system recovery and non-recovery. And this was work done with high content imaging data that was concentration, response, and time course data in HepG2 cells. We developed a methodology that I'll briefly talk about in a second. And then we applied this method to a very different model of neuronal networks, rat hippocampal neurons, and measuring their perturbation by chemicals over time using microelectrode arrays. And then the last model we looked at was induced stem cells and transcriptomics, where we again used the same methodology that we had developed for high content imaging and applied it to stem cells and tried to look at tipping points and differentiation trajectories. So all of these studies treated cell culture models with different doses across time points and then measured the perturbation using multiplexed assays. And the idea of finding the tipping points, the key concept was that each assay could measure the perturbation of the system at different chemical doses and follow the trajectory of the system over time. So these graphs that are shown on the left-hand side of this slide show trajectories over time that are idealized trajectories that capture the overall perturbation of the system. So at a very low concentration of a chemical, you may observe no effect or no significant perturbation. With an increasing dose of a chemical, you might see some sort of adaptive change that leads to changes in adaptive stress response pathways, mitochondrial functions. But then over time, you see recovery of the system back to some basal level. But then there's this critical concentration of the chemical that is right at this tipping point. And if you exceed that concentration, what you find is that the system doesn't actually recover over time. So this early notion of a tipping point was based purely on the idea of following the system over different concentrations in time and trying to estimate cellular recovery. As we move forward now and look at more cell-based data and transcriptomic single-cell data, we've been thinking more about, can we expand this notion of tipping points to not just this notion of recovery, but as thinking about the system in terms of cell states and cell state transitions. So imagine under the normal conditions, a cell would be in a given state of function, whether it's dividing or metabolizing nutrients and just growing. And if you expose it to a certain concentration of a chemical, it might undergo some adaptive change. It might induce enzymes. It might result in producing reactive oxygen species that cause activation of oxidative stress response through NRF2 and KEAP1 that induces certain enzymes that result in, again, recovery of the system from whatever the perturbation was back into the normal state. But then if you continue to expose the system to a increasing dose of a chemical, you might exceed some threshold that prevents the system from recovering. And I'm really glad to hear this, that one of the speakers, in fact, or one of the participants in the call was, in fact, talking about the same exact situation. So there's this theoretical threshold that exists out there in cells, that if you exceed that, you can go into some adverse state. And in the case of, again, single cell responses, we can think about these adverse states as being potentially apoptotic states, necrotic states that result in cell death, even autophagy. So this is the sort of new idea that we've been leaning towards now, is thinking about tipping points as critical thresholds that you exceed. And as I mentioned, adaptive stress responses are really, we think, key to understanding these and estimating and using these tipping points. So chemical exposures can trigger adaptive stress pathways within cells, activate detoxifying enzymes, stress proteins that counteract the effects of chemicals. And this has been summarized very nicely by Steve Simmons in 2009, where he's talked about this sentinel cell stress responses, including oxidative stress, heat shock response, DNA damage response, hypoxia, unfolded protein response, and other modules that are involved in cellular adaptation to chemicals. So as long as the chemical exposure is low, adaptive stress response pathways, if they're not overwhelmed, you get recovery, and if they are overwhelmed, you can cause cellular dysfunction, damage, and various disease states. investigating is can we use single-cell transcriptomics to identify these tipping points? And the hypothesis is that these tipping points are thresholds between heterogeneous adaptive and adverse cell responses, and that you can use single-cell transcriptomics to identify these. And the approach that we're using is we're starting out with a set of reference perturbogens that are well known to activate specific adaptive responses. We're using a model of liver-derived cells, HEPA-RG cells, which we have a vast amount of data on already in bulk transcriptomics, as well as there's a lot of liver biology that we can bring to bear upon this problem. And so the challenge is, how do we generate the single-cell transcriptomic data? How do we analyze the single-cell data to identify these different cell phenotypes? And then eventually, how do we use systems modeling and virtual tissues to simulate the cell trajectories that represent these normal perturbations, the adaptive changes, and then adverse effects that cross the tipping point. So just as a background, heterogeneous cell responses are really critical for understanding health and disease. There's a vast amount of literature in toxicology on the liver in terms of diverse cell populations, hepatocytes, Kupffer cells, stellate cells, and how they respond differentially to different hepatotoxic agents, leading to varying degrees of injury, fibrosis, carcinogenesis. The same is true for the kidneys. Nephrotoxic effects can happen in proximal tubular cells versus podocytes. They can have different impact on kidney functions. In fact, there's another project currently ongoing in the virtual tissues led by Tom Knudsen, in which the differential effects on neuronal and glial cell populations is being investigated to try to understand its impact on neurotoxicity. So, in addition to tissue-level responses, there's also a great deal of variability that can happen within a single cell type. So the variability in stress responses being the case in point, hepatocytes can show differences in stress response pathways, and those can lead to toxicity as well as disease. And there's a huge amount of literature growing on cancer cell resistance, on how tumor cells show heterogeneous cell autonomous responses to chemotherapy, and that are really the subject of single cell analysis approaches. So single cell analysis approaches have really been growing over the last 5 or 6 years, and there's tremendous utility in applying them to various diseases and hopefully as well to toxicology. So one of the things that we've been doing is we've been building a database of stress perturbogens or stress inducers from literature. A lot of folks like Chris Corton and others have in fact started this effort out by capturing very wonderful articles on DNA damage response inducers, oxidative stress response inducers, and unfolded protein response inducers. And we've taken those papers and other review articles and assembled a large data set of reference perturbogens for which there is significant evidence in literature for the induction of these adaptive stress response pathways, including the DNA damage response, heat shock response, hypoxia, metal stress, oxidative stress, and unfolded protein response. In addition to gathering evidence on about 100 or so chemicals, benchmark chemicals, we've also developed the text mining approach. This is work done by Brian Chambers, and this is currently in review. We've developed a database of 500 reference perturbogens that are mostly out of the LINCS database, out of Broad. and we're currently expanding on this set. These chemicals activate the adaptive stress response pathways, and we've curated evidence about them out of a couple of thousand articles. Hopefully this data set will be available to everyone to use for various analyses. So in addition to building a database of stress chemicals, we're also trying to build an automated approach to generate accurate signatures, gene signatures for adaptive stress response pathways. This work was again done by Brian Chambers, and he used literature evidence on a subset of chemicals that were available to us 2 years ago, and created signatures for DNA damage response, unfolded protein response, heat shock, hypoxia, metal stress, and oxidative stress. One of the challenges that continue to be an issue for analyzing the data is that there's a lot of crosstalk between adaptive stress response pathways, and building signatures that are completely unique to a particular type of stress, A, may not be possible, and B, may not be useful because there's a progression of events that occurs that relates these stress response pathways. But more about that as I share more data with you. So single-cell RNA-seq is really evolving very rapidly. Now I'm going to act like I'm an expert in single-cell RNA-seq, but I'm not. I have a really smart bunch of collaborators, including Brian, David Gallegos, as well as Dennis Eastman out at BioSpider, who've been really helpful in educating me about the different types of technologies that are available to us. So the number of publications are doubling every 5 years. About 5 years ago, there were about 800 or 900 papers in PubMed, and now there's 5,000. One of the key challenges for us is that the assays and sequencing are really expensive. I remember about 10 years ago when we were thinking and wishing that we could run single-cell assays, the cost per cell, the cost per sample was astronomical. We couldn't run a lot of samples, and the data was really noisy. Now, fortunately, with time, the technologies have several technologies are available to us. So we're evaluating 10x Genomics, Parse, as well as an offering from BioSpider that I'm going to jump into much more detail. So we've been using the BioSpider TempoSeq gene expression assay for several years now, generating, as many of you in the audience have probably been using data from generating TempoSeq data as well. TempoSeq has many advantages. There's no cDNA synthesis. There's no preamplification required. You can work straight off cell lysates. And there's a targeted transcriptome or a whole transcriptome that you can use. You can attenuate transcripts so your read space is not chewed up by mRNA that you don't really care about. This lowers the sequencing costs, and the analysis is really straightforward. So it's a really powerful alternative to doing traditional RNA-seq, and we've been using the data for several years, as I mentioned, and had a lot of luck with it. So naturally, we were very keen to see if BioSpider could have some sort of single-cell offering. Unfortunately, they were just working on a technology to enable just that. And so the TempoLink platform is the single-cell genomics platform that basically is an instrument-free single-cell genomics platform that combines a targeted approach that can look at any number of transcripts up to the whole transcriptome, produces really high-quality data, and currently you can look at 48 samples, which is what we did. It's really scalable, as you'll see from the number of chemicals and cells that we ran through this assay. The barcoding process is based on split pool approach. So you take the whole transcriptome primers, and then you index each of the samples, and then you apply various barcodes in a split pool paradigm to be able to scale up to about 1 million cells. There's about 2 million unique barcodes, and the potential for collisions between these barcodes is really, really low. So this approach has been compatible with Illumina sequencing, so it's easy for us to basically send samples to them. They can barcode them, prepare them, and then send them back to us for sequencing. So in this process, we've been developing a workflow, which I must say it's been a lot of hard work trying to figure out some of the early steps, but it has come together really very smoothly. So the early parts of this process are cell culture, chemical treatments that happen over here at the EPA, cell preparation, cell fixation, cell dissociation, again happens at the EPA. And I got to thank Brian Robinette for all of the upstream work and labor on getting all these cell cultures ready. David Gallegos spent a year of his life trying to figure out the cell fixation and dissociation protocol, and gave me a newfound respect for the kind of work that you have to do in the lab to figure out how to separate these cells. Separating these cells out after treatment and making the pellets, those are shipped off to BioSpider, where they do the permeabilization, the detector oligohybridization and barcoding, ship back the samples for sequencing. And in our case, we were fortunate to have a collaboration with Doug Bell at NIHS and the sequencing core through an IA, and we were able to get all the sequencing done with the help of NIHS, for which we're very grateful. The data was sent back again to BioSpider. They demultiplexed it, filtered out the cells, gave us the counts with all the QC. We normalized the data, batch corrected it, and did the analysis that I'm going to talk about. Bryant Chambers, who's no longer here at EPA, but has now moved on to Unilever, has been closely involved in this analysis, as is David and myself.
I think we just lost your slides. Or is that it?
Can everyone see my slides?
I cannot. Can anyone else confirm if that's just me?
Yeah, I don't see the slides.
Yeah, I can't see them.
Give me 1 minute. There we go. Are we back?
Yes, looks good.
Excellent. Nothing like having technical problems. All right. So the cell culture and treatments, we picked a handful of chemicals because it was a pilot, and truth be told, like most pilots, we did not know whether this would work or not work. But we selected a set of chemicals that were well-known stress perturbagens. We picked Brifaldin A, picked cytotoxic doses. We checked the viability to make sure that not too many cells died. DMSO was the control. For DNA damage response, we picked etoposide. Oxidative stress response, we picked TBHQ, tert-butyl hydroquinone, which since then I've been told by several colleagues that may not have been a great idea, since TBHQ does have a habit of degrading, and maybe we weren't actually seeing true effects. As positive controls, we had rotenone and cyclohexamide, mitochondrial stress and protein synthesis inhibition. And then for good, just for fun, we also threw in a triglycerin, a PPAR alpha inducer, again as a reference chemical, to see just what you might see. And for fun, we tested it at doses about 10 times lower than what we had wanted to. We had thought we might test it up to 10 micromolar, but the dose range got shifted by 1 decimal point. But we found some really interesting results. And lastly, we ran tunicamycin again to look at unfolded protein response. The samples were, again, prepared as I described earlier, fixed, dissociated, sent off to BioSpider. They put the samples through their workflow, and voila. After a lot of hard work by a lot of people, we found this really beautiful data quality plot. So what you're seeing on the left-hand side, if you're familiar with single-cell transcriptomics or single-cell RNA-seq, is what is known as a knee plot. And what it's showing on the x-axis are the number of barcodes, which correspond to the number of cells, and then read counts on the y-axis. And essentially what you look for is this peak in the number of barcodes that corresponds to this knee right over here, which corresponds to actual cells. Points over here, up here, are generally broken down cells that have high, small numbers of barcodes and high numbers of read counts, but not actual viable cells. So this knee plot is what tells us whether you're getting in actual cells or not in your assay. And what we found over here, and actually this is from BioSpider, thank you, that there were approximately 74,294 hepatocytes identified, or cells, I should say, identified at least at 10,000 reads per cell. The read depth was anywhere about 10,000 on average. We had really high map reads, so all the reads that you can see on average 87 to 90% of the reads were actually mapped to actual genes. We also had a linear relationship between the UMIs and genes, knowing there was no saturation going on, so we could be, again, the data looked really clean. Once we got the data from BioSpider, we basically used open source tools to analyze the data, processed it through this package known as ScanPy. Some of you may be familiar with that. It really makes an easy job of doing large-scale analysis. So we had 74,294 cells, 22,533 mRNA probes. And just to pause there for a minute, the entire large-scale HEPA-RG screen that we ran with 1,500 or 1,800 chemicals had fewer profiles than this single 48 sample single cell transcriptomic study. So it was quite a bit of data to deal with. We filtered out the first analysis I'm going to show you today is going to only be the 24-hour data. And just to keep the quality really high, we just picked only those cells that had at least 1,000 genes and at least 1,000 counts, reads in them, which left us with 33,742 cells. We normalized these counts per cell to 10 to the power of 4 counts, log2 transformed it, and then had to batch correct this data because there were a number of batches just because of the processing. And it's always critical to oh
no, did we lose Imran?
I think he's back.
He's back.
I thought I saw the slides.
I have his slides as backup, but I don't have him, sadly. Did we lose him?
Looks like it.
There he is. Imran, we lost you for a second.
I am back.
Okay.
I'm back. I'm very resilient. I keep coming back. So, okay. So we batch corrected the data, and the first thing you want to do is figure out if you have clusters that are meaningful. Are the cells that you get out of this study, if you group them together, do they correspond to the actual treatment that you applied, and does it make sense? And when we saw this result on the left-hand side, we were really excited because all the chemical treatments formed really nice clusters. But on further examination, we realized that that there was some batch effects going on, and it really doesn't necessarily make sense for each chemical to uniquely cluster into its own clump, because that means that there's no common biology happening between those potential cells. So in any case, after doing a batch correction using the harmony approach, which is shown to be more robust than some of the other approaches, we found the grouping on the right-hand side. And what you're looking at over here are T-SNE plots. The colors correspond to the different chemicals that are shown over here. DMSO control, bromelain A is blue, DMSO control is purple. Cyclohexamide is this bluish tinge, light blue, more Carolina blue. Etoposide is more of an aqua. Rotenone is greenish. TBHQ is orangish. Troglycazone is yellowish, I beg your pardon. Troglycazone is orange, and tunicamycin is red. So you had tunicamycin falling out right over here as its own cluster. But when you do batch correction, what occurs is that the DMSO area, the DMSO clusters are right over here in the middle where my mouse is right now. And the low doses of the chemicals, as you'll see in the other results, tend to group over here in the DMSO cluster. But then the higher doses of bromelain A and tunicamycin, they're right over here. Rotenone is up here as well. Cyclohexamide and etoposide high doses are over here. So there's some pretty interesting structure over here. And so this was encouraging in a way, but we still had to figure out, what does this mean biologically, and are we seeing adaptive stress in any way? So before I jump into telling you about some of those stress responses, what I wanted to say something about, a comparison with bulk tempo-seq data. It's instructive to think about, what can we get out of this sort of assay that you couldn't get out of bulk data. So what I'm showing you here is 20-odd replicates of troglycazone that we ran in our bulk HTTR assay with tempo-seq, and differential expression was calculated. Comparison DMSO controls. This is the 24-hour data, 0.01 to 100 micromolar treatments. We did concentration response analysis for a whole host of troglycazone PPAR alpha targets. I'm showing you fatty acid binding protein results because that seems to be the most robust response in this data set. And what you're seeing in this heat map are actually heat maps, concentration response curves, basically stacked up, where the red saturation is showing you log2 fold change induction, and the concentrations are shown on the y-axis. So one of the things that's pretty evident is that below 1 micromolar there's almost no significant effect, right? So this is what we have in the bulk data, and if you go to the single cell data, even though we ran troglycazone at a maximum concentration of 0.1 micromolar, we had 3,849 troglycazone treated cells in this data set at 24 hours. And if you look at the distribution of, again, this t-SNE plot that you're looking at over here is showing you the distribution of cells with the different markers for or targets for PPAR alpha. So you've got HMGCS2, you've got fatty acid binding protein, CPT1A, PDK4, CREB3L3, and CYP4A11. CYP4A11, this probe may not have been great in this particular case, but again, you see some subset of probes are turned on. Again, for HMGCS2, you can see a whole number of cells are showing activity on a background of very little activity. And fatty acid binding protein, there's clusters of responses, cells over here that are actually showing quite a bit of activation. So if you look at the distribution of up here on the right-hand side, in this graph, you're looking at the distribution of log2 fold change values. So this is log2 fold change calculated by comparison with DMSO treated cells only. And there's many ways in which one can actually do this analysis, but this is just based on the DMSO comparison. And what you're seeing is the distribution of log2 fold change values for these 3,849 cells at different concentrations. So the first group over here is the 0.001 micromolar concentration. This is the 0.01 micromolar, and this is the 0.1 micromolar concentration. Substantial responses, right? And if you look at the number of chemicals the number of cells that are greater than a log2 fold change of 1, you see a nice dose response. So there's a lot of sensitivity in this data, something to think about. You can see effects at 0.01 and 0.1 micromolar that are absolutely missing in any of the bulk data. So that's just the smallest side to signify the sensitivity. Moving forward, the next thing we wanted to see is, well, what do the cell responses actually mean? And can we cluster them phenotypically in some meaningful way? So there's a variety of algorithms that you can use. One of them is this community detection algorithm that you can, that basically look at the similarity between profiles and try to group the chemicals together. There's many parameters that you can set on this. You can get a small number of clusters or a larger number of clusters. And again, the results that I'm showing you are based on a particular value, R value of 2 for the Louvain cluster community detection algorithm. And what you see, each of these numbers signify a community or a cluster. They're also shown in colors over here and in numbers over here. The distribution of cells in these clusters corresponding to all the treatments are shown in the heat map on the right-hand side. So the first row in this heat map shows you the distribution. The saturation over here is the count of cells in each cluster. So DMSO has a very characteristic distribution across cluster number 2, 3, 5, very few or none in 6, 7, then 8, 9, and then some in 15, 16, 17, and 18. If you look at Brifildin A at the 0.001 micromolar, the distribution of cells for Brifildin A for this low dose look really similar to DMSO, right? It's almost the same. So as you increase the Brifildin A dose, you see that cells pop up from this normal DMSO treated distribution to a slightly different distribution. Cells, again, they go into different states. At again, a higher concentration of 0.1, you see again a slightly different shift happening. So again, there's shared clusters between the different treatments, and there's some unique clusters as well. Rotenone produces some really unique clusters over here. And so the next question for us was, okay, what do these clusters mean, and do they make any sense with respect to the biology that we expect to see? So what you do next is that you go through each of the clusters and you use an approach to try to find what are the key differentially expressed genes in each cluster by comparison with all the other clusters. And there's, again, many ways to slice this and to calculate these DEGs. So we just took a very simple approach, and the graph that you're looking at over here is a dot plot showing you on rows what the clusters are, and on columns they're showing you genes, and this is just the top 3 genes out of each cluster, just for the purposes of showing you intuitively how this process works. So cluster number 9 has these genes that are really differentially expressed in it. Cluster number 11 has these. So you can get a differential expression profile for each of the clusters, and from that you can then figure out how you're going to do the enrichment analysis or analysis of any other signatures. So for this analysis, then we took 100 to 200 DEGs out of each cluster and tried to figure out what the enrichment was. So we did a bunch of analysis with MSigDB signatures, hallmark signatures, canonical pathway signatures, which were interesting. We got some pretty interesting hits from those that we would expect. But we also took the DEGs and compared them with the adaptive stress response signatures that we published a few years ago. We actually expanded the set of signatures to not only include unfolded protein response, oxidative stress, metal stress response, heat shock response, hypoxia, DNA damage response, but Bryant also took a similar approach and built this consensus signatures for apoptosis, autophagy, senescence, as well as lysosomal function. And one thing that he's always been really interested in is aggregosome formation, which is a step in UPR. Hopefully someday he can talk about that to everybody in more detail. And then the question was, how do we match these clusters and how they align with these adaptive stress responses and link them up with the chemicals? So we basically just looked at cluster by cluster, where were the p-values below the threshold of 0.01, and line them up with these clusters over here for each of the chemicals. And what we found was that on the left-hand side, I've shown you some of the treatment concentrations for some of the chemicals. And on the right-hand side, I'm showing you the matches that we found between the different treatments and the adaptive stress responses. So let me start with Brufel A. So Brufel A, at a dose of 0.1, there was enrichment, significant enrichment for UPR, as you would expect. For etoposide, we found at different concentrations enrichment of DNA damage response, as well as apoptosis at the highest dose. For rotenone, we found hypoxia. We also found hits with rotenone as well as some hallmark pathways. Since we haven't actually covered all of biology in these gene signatures, there are other hits that we get to these clusters that make sense biologically, but just in the interest of keeping it simple, I'm showing you some of the illustrative examples. Again, with tunicamycin, we saw hits on unfolded protein response. We also saw other hits on some hits, low hits on DNA damage response. UPR was actually divided across different clusters, and there were slightly different sets of genes in those. And we're still trying to work out which phase of UPR these particular clusters may correspond to. TBHQ, we didn't get great hits for oxidative stress signatures. That's been one of the challenges that we had even in the bulk data. We just didn't find great hits for oxidative stress response that we would expect to see. And I don't know if it's an issue of us not being able to build the right signatures, or just the key genes that are not expressed in that way, or it could be some other technical factor that we're not aware of. But this was actually encouraging, because what this suggests is that you can actually use single cell approaches to find these responses, to find actual cell states corresponding to adaptive stress. And of course, we're not done with this, and this is still a work in progress, but you know, it's very promising in that regard. So how do we find tipping points? And what we're thinking is that the reference chemicals seem to induce the adaptive states that correspond to known mechanisms. That's not precisely, but there's a decent match between them. One thing we find is that you get this big blob of DMSO normal cells over here and over here that when you increase the dose, for instance, of Brufel A, the lowest dose of Brufel A, cells are mostly sitting over here. When you hit them with a slightly higher dose, they jump up, and many of them jump into this adaptive state over here, along with tunicamycin as well. So could this be an adaptive state? Potentially. And then this cluster over here of cell states corresponds to cyclohexamide and rotenone. And based on some of the analysis, not the apoptotic signatures that I showed you, but other signatures of apoptosis, we think that there's higher chance of there being apoptosis over here. So there seems to be a trajectory or a potential pattern that goes from normal to adaptive and to adverse in this particular direction, at least one of the trajectories. And so one of the questions, as you are well aware of, you can use single cell data to in fact infer trajectories. So one of our objectives is next to identify potential trajectories out of these data. But also, one of the things that we may be missing over here is the time points. So we missed out potential early responses since we just looked at 24 hours. And even though we have 3 doses, we may be missing the early critical steps that may go and connect some of these cell states that are here in this normal stage with the adaptive stages, as well as going higher in dose. Maybe we need to push the cells a bit harder with Brufeldin A and tunicamycin to see them push towards these apoptotic outcomes, or even see if we can find autophagy. There is some indication that we may have some autophagy in this data set, but again, that requires further work to determine. So what I've shown you over here is that we can use single cell data to identify cell phenotypes, and they hold the clues for the critical transitions that may allow us to figure out how cells go from recovering into potentially cell adverse states, apoptosis, autophagy, or senescence. Of course, we need to do much more experimental work and much more analysis. And of course, I'm open to, if you have any ideas on how you think we might approach this, be happy to get your input. So, very brief summary. So we think single cell transcriptomics is a really powerful tool that can enable us to decode heterogeneous chemical induced effects of specific as well as potentially non-specific chemicals. We need a scalable tool platform that can generate such data. We think TempoLink is a pretty effective workflow, and the data format is easy to use, and there's open source tools out there that can help you dive into analyzing these data. Of course, there's many technical issues that still need to be addressed and resolved. And we're thinking that these data could provide very useful clues about studying adaptive states and transitions into potential adversity. I think they'll take a lot more modeling and analysis and simulation, as well as biology and biological insights to be able to solve these problems. And then thinking about whether and how these might be relevant for risk assessment, and what kind of evaluations and validations would be necessary to take them to the next level where they might be useful. So then I'd like to thank everyone who did the work. Brian Chambers, postdoc, who's now at Unilever. Brian Robinette, who works with Brian Chorley, who's done a lot of work and deserves a great deal of credit. David, who again put in blood, sweat, and a whole year trying to figure out how to fix and dissociate these cells. Tom for his input on the project. Brian for being a great collaborator and making it all happen. I'm not a wet guy, and so he's been able to make this all happen experimentally. Biospatter team, Dennis, for all the leading the protocol, the TempoLink platform, and walking us through how to basically use it. Bruce for his insight, and Joel for being a great team player, and for his insight. The NIHS sequencing core was again critical for making all the sequencing happen. Doug Bellinozzi was involved in the early stages of this project and were instrumental in getting this work done. And Alistair out of Unilever is a collaborator who I keep talking to and bouncing ideas off on adaptation and adversity and how we can generate the right data sets to model them to be able to predict these tipping points. So with that, I'll thank you and take any questions.
Thanks, Imran. I know we're a few minutes over, but we have some time baked into the end of the meeting, so I promise none of the work groups will lose their time. Peter, is your question related to this? I see your hand up first.
Yes.
Yeah, go ahead.
So I have a question. So I noticed that there are 3 distinct clusters with the cyclohexamide. So is that because instead of a normal versus damaged or whatever, just a simple threshold, that there's maybe different states that can go from the normal state to that you have different responses, that the cell is somehow making decisions as to how to respond to this stressor?
I think that's probably a fair. So cyclohexane.
Go forward a couple. Keep going. There. Look at the three at least three different green groups.
Yes.
So is that it, or is that explained some other way? I'm not sure how to interpret that.
So there's a lot to unpack over here, and I have not done justice to all the potential cell states that are available here. So this is a protein synthesis inhibitor. So you're actually going to see impact on protein translational inhibition, and some of the effects are just protein translation that nothing else is really impacting in a way. It's really unique to that. One of those clusters corresponds to, and I haven't shown you, in fact, all the treatment concentrations for cyclohexanide here. You're only seeing one of them. The three others show you at very low doses other areas where they're active as well. So there's really a lot of heterogeneity in the kinds of things that are happening to HEPA-RG cells. Protein translational inhibition is one. Cell function, cell cycle is another. And then once the cells actually start getting damaged, they look a lot like rotenone. And this cluster, my speculation is that this is apoptosis right over here, or pre-apoptotic cells.
Quick follow-up question then. Does that follow like in a straight trajectory when you do the trajectory analysis, or is that like a branch trajectory?
I couldn't speculate, and it could be either. Mathematically proving that it's branched or linear is very difficult with these data because of the nature of the connectivities between the cell states. But if you're interested, please follow up, and we're happy to look at this further with you.
Okay, thanks.
Great
talk,
Peter.
By the way.
There's a couple of questions in the chat. Some are related to cell type. So one is, have you used this with a mortalized cell line? And another question, have you considered using co-cultures for things like immune cells?
This, other folks can probably chime in and give much more information about this, as I haven't tried it with many other cell types. We actually were supposed to have two cell types here. Cholangiocytes, and I'm sure other people have questions. There should be cholangiocytes in here and hepatocytes in here. We have looked with a fine-tooth comb for cholangiocyte markers and haven't found them. So if you have brilliant ideas of cholangiocytes and how we find them, it could be that they didn't make it past our dissociation protocol.
Yeah, Imran, really great talk, and congratulations on what you've done so far. It's really fascinating, and I think it's going to be very useful. I wonder, though, in the cell cultures, that when you get to a point where the cells are basically never going to recover, are you going to see those cells at all? And I think this relates to what Scott has in the chat. I'm just wondering if they will just be that you won't get a good single cell sequencing of those cells that are basically dead or about ready to die, right?
Yep, you're absolutely right. There's a lot of dead cells that are gone, so we've selected against dead cells, and so probably what we're going to find are pre-apoptotic cells potentially, and if that I can't be sure. The signal for apoptosis is not strong, so there's a good chance that we've lost them. So that's another reason why we want to go back and look at earlier time points and see if we can get a slightly stronger. I mean, 24 hours. What do you think? Is it too late? Because we have a 48-hour time point as well. When would you see a pre-apoptotic signal transcriptionally in these cells, do you think? Would it be 12 hours? Would it be? I'll take a vote.
Yeah, do a time course and find out.
Yes.
Yeah.
Okay. Scott.
I was just wondering about selective pressure in a culture environment, right? So you push cells to an adaptive state, and there's some selective pressure for which ones that respond and which ones don't. So there's an epigenetic landscape that's somewhat variable depending upon cell to cell, right? And so you're selecting out certain ones to move out into that space, right? So you're basically imbalancing the culture system at that point based upon the chemical exposure. So this idea of adaptation and then full reversion, so when you take the chemical away, considering you've put on some selective pressure and changed the distribution of the epigenome probably to some degree in a component and subset of the cells, do you ever think they return to the actual original state from where they came from?
I used to want to believe that. Theoretically, it's a beautiful idea that they recover, isn't it? Isn't it beautiful that they recover? But I don't think they can, because there's memory, right? And they, you know, so there's changes. You're right. Epigenetic changes have happened. Transcription factors have been, chromatin's opened up, and there's memory that has to be retained. So probably they go into some sort of what is that called? The place in between heaven and hell.
Purgatory?
Purgatory. They're living in purgatory.
Very good.
Probably not ever coming back to baseline.
Gotcha.
Limbo.
I guess there's a functional question too, though. So they get roughly functionally back. Like Dave Malarkey did studies years ago where he used non-genotoxic chemicals to induce tumors in livers and then took the chemical away. And the chemicals were dependent upon the tumors to drive their growth, and they regressed. And so there's some of that, but yeah.
Well, hopefully we can test some of that in the 3D hepology 3D model a little bit more, because they're more resilient and maybe they can withstand this a little more. But
all right, let's do Leah's as the last one. So Leah asked, how does the tipping point fit with a cytotoxic burst absorbed at higher concentrations with cell exposures? Would you say this burst is a biological tipping point in cells, or does it occur lower than the burst?
is Richard on the phone? Is Richard here or not? If he's not
Richard
here, no. Richard Judson. So the cytotoxic burst, we've had many debates about that. And based on the data that we saw for the tipping point analysis in HepG2 cells, and even in neuronal cells, we found that these tipping points were generally 2 to 3-fold or more lower on average than cytotoxic burst concentration. So they're really preceding any kind of burst of cell death. Now, is it related to stress? Could it be a more downstream part of the cell not recovering and then going into death? Potentially. But based on the data that we've seen and analysis that we've done, they precede that, and they're more sensitive than a cytotoxicity.